Hello, everyone. I'm Susan Schantz. I'm the director of the Children's Environmental Health Center Research Center at Illinois. I want to thank everyone in the audience today for coming to our first annual Midwest Children's Environmental Health Symposium. I especially want to thank our colleagues from the Children's Health Center at Michigan for joining us today. This is the agenda for our session. We're going to first hear from some speakers that will talk about the research and outreach going on at the Illinois Children's Center. Then we're going to take a break and go out into the atrium of the Beckman Institute from 10:30 to 11:30. We'll have a poster session. Students and postdocs from both of the two Children's Center will be presenting their latest research. Then we'll reconvene from 11:30 to 1, and we'll hear from speakers who will tell us about research and outreach at the Michigan Children's Center. So I wanted to start today by just introducing you to the Children's Environmental Health Research Center's program. This is a program that was actually initiated 17 years ago. It's a little unusual in that it's a joint effort between two different government agencies, the NIEHS, or National Institute of Environmental Health Sciences, and the U.S. Environmental Protection Agency. And throughout the entire length of the program, these two agencies have collaborated to jointly fund each of the children's centers. It's a network of research centers that was set up to study the impact of environmental factors on children's health. And this slide here shows all of the current centers that are funded. There are currently 14 children's environmental health centers at academic institutions across the country. As you can see, Illinois and Michigan have the distinction of being the only two centers in the Midwest. So then I'd like to move right along and introduce you to the Children's Center at Illinois, tell you a little bit about the focus of our center and how it's organized, and then you'll have a chance to hear about the research that's going on in the different projects within the center. So our focus of our center here at Illinois is to study the impact of chemicals that are found in consumer products on both reproductive and neurodevelopment. And this slide illustrates some of the key chemicals that we're focusing on. Parabens are used as antimicrobial agents in a number of different kinds of personal care products like lotions and makeup, for example. Phthalates are used in many products, but one of the primary uses of phthalates is to stabilize the scent in various types of scented personal care products such as shampoos, lotions, some makeup, perfumes, etc. Bisphenol A, or BPA, is used in polycarbonate plastics that are used for as food containers and also in food packaging, and importantly in the lining of most metal food and drink cans, including your Coca-Cola can, as you see here. And finally, triclosan is an antibacterial agent, so it's the agent that's found in those antibacterial soaps that you see throughout your house and in many bathrooms in public places. And it's also used in some toothpastes and mouthwashes. So these are the chemicals we're focusing on. Our center is assessing exposures to these chemicals at two critical periods of development, the prenatal period and also the adolescent period. We're investigating as well whether obesity increases the risk from exposure to these chemicals, whether it's maternal obesity during the prenatal period or child obesity during the adolescent period. And then we're investigating the role of oxidative stress and inflammation in mediating the outcomes of interest. This slide lists the various center investigators that are involved in our center at Illinois. I'm not going to go through and introduce every person individually, but I just wanted you to know that it's a very diverse team of people with many different scientific backgrounds that are collaborating on this research. and that some of the faces you see here will reappear as I tell you about the work that I'm involved in. This slide lists the projects and cores for the Illinois Children's Center. We have 3 research projects in the center and 2 cores. The first project is human studies. The title is Joint Effects of Endocrine Disruptors, Diet, and BMI, Body Mass Index on Child Development, and that's led by Susan Korick and myself. And this project includes both a new prospective birth cohort study in which we're recruiting pregnant women and following their infants prospectively, and then also a cross-sectional adolescent study that we are embedding within an ongoing prospective birth cohort study that Susan Korick has been involved in for a number of years. And she'll tell you about that study in a bit. Project 2 is titled Endocrine Disrupting Chemicals, Diet, and Gonadal Toxicity. And this is studies done in mouse models. Jody Flaws is the lead for that project. And then lastly, Project 3 is Endocrine Disruptors in Diet, Effects on the Developing Brain or Cortex, and that's led by Janice Duraska. We have a community outreach and translation core, and community outreach and translation is also actually a really important part of all of the children's centers throughout the program, and Barbara Fees is the director of that. And then, of course, we have an administrative core that coordinates all of the activities of the center. One thing that's unique about our center is that we are attempting to conduct parallel studies in animal models and exposed human populations. So we have tried to put very careful thought into how we could model exposures in the human population in our animal models. For example, in our studies of phthalates, we have looked at the profile of phthalate metabolites in pregnant women in our birth cohort, and then made up a phthalate mixture in the proportions that are found in the women so that we can closely model the mixture of phthalates that women in our studies are actually being exposed to. We're using the animal models to conduct studies that will probe the underlying biological mechanisms of these chemicals, and we're also using them to generate results that will guide the selection of health outcomes in the children as we follow their development. So I'm going to spend a little bit of time now telling you about the new prospective birth cohort study that we're conducting here in Champaign-Urbana. And an important goal of this study is to develop better tools that will more reliably identify neurodevelopmental risks from prenatal chemical exposures. And to do this, we're trying to use tests that assess core cognitive abilities, things such as information processing ability, working memory, and attention. And we've selected this approach because these types of basic building blocks of cognition tend to be stable within individuals over time. So we hope that if we can measure these accurately early in infancy, that this will predict whether there will be lasting deficits from chemical exposure. And we hope that by developing these procedures, this will allow for both at the individual level for earlier interventions to help reduce risk, and at the population level to more timely regulation to protect children's health overall. So this is a pregnancy timeline that shows our data collection process for this study. The study is called the Illinois Kids Development Study, or IKIDS for short. And we are recruiting women in the first trimester of their pregnancy, around 10 to 14 weeks of gestation. And then we see them periodically throughout the pregnancy to collect both exposure data and covariate data. And I'm not going to go through all of the different assessments we do at each of these time points, but this is shown on the slide. Our goal is to enroll 600 women, and our current status is that we have about 273 women enrolled, and we've had 187 births. So we're approaching the halfway mark in recruiting our cohort. This slide shows the infant assessment timeline for the iKids study. So you can see that our study is very heavy on collecting lots of data early in postnatal development. So we see the children at birth, where we collect physical measurements, a cord blood sample, and meconium for microbiome studies. We see the babies for the first cognitive assessment between 1 to 5 weeks of age, where we're looking at memory and attention. We see the babies again at 4 to 5 months, where we're looking at physical reasoning, mental rotation. We're repeating the physical measurements we did at birth, and again collecting stool samples for assessments of the microbiome. And then we see them again at 7 to 8 months of age, where we repeat memory and attention tasks that are very similar to what we did in the newborn period. We have a theory of mind task that I'll tell you about, and we're also looking at event-related potentials that we hope will give us some clues about the underlying functioning of the brain that might relate to the cognitive effects that we're measuring. So in this slide, I'm going to tell you a little bit about the methods that we've developed for assessing memory and attention in very young infants. And this is due to the work of Andrea Aguiar, who's a member of our center research group. She's a developmental psychologist who focuses on infant cognition. She's very, very knowledgeable about very young babies and how they think and what they can do. And so we've actually gone through a few prototypes to get to the way that we're now assessing the infants, and I think we've got a design that's pretty amenable to getting good data from very young infants. So this is, on the right, you see Kelsey, who's a neuroscience student working on the project with one of our study participants. And you'll see she has the baby in a harness, and that allows her to hold the baby upright and keep the baby positioned appropriately to look at the stimuli on the test screen. On the top part of the apparatus, Kelsey's looking at a video image of the baby. You can't really see it, but the baby has a little target sticker on his forehead so that she can make sure that he's aligned to the center of the apparatus. And right near the baby's forehead, there's a little headrest. And when the testing starts, she's going to gently place the baby's forehead right against that headrest. And that ensures that the baby's the right distance from the screen so that to be able to see the stimuli well. Babies this age can see, but they can't see very well. So you want to make sure that you have them the right distance from the stimuli. On the other side of the apparatus is Francesca. She's another neuroscience student working on this research project. She's looking also at a video feed of the baby, and she's going to operate the testing. So everything is computer controlled. Once she sees that the baby is awake and alert and looking to the center of the stimulus display, then she'll start the task. And from that point on, everything will be computer controlled. And what she'll be doing is just using keys on the computer keyboard to record whether the baby's looking right or left, to the center, or off the stimulus display altogether. So now I'm going to illustrate for you what the baby sees. So initially the baby sees this cute little green guy in the center of the display. And this is an attention-getting stimulus to get the babies focused on the center. So the baby hears this little guy saying, "Baby, look! Baby, look!" And once the baby has looked for 2 seconds, then the stimuli will be displayed. And there are two different tasks. Here I'm illustrating one with faces, but we also do one with geometric figures. So first the baby sees two identical stimuli, here two human faces, and is allowed a period of time to study these stimuli. And Francesca is recording the direction of the baby's look and the length of each of the looks. Once the baby has habituated or become very familiarized with these faces, he'll start to look away more and more and have shorter looks. And at that point, we'll go on and present a test trial. So the baby will again see the little green guy, and then once he's paying attention, then he'll see the familiar face with a novel face. And the typical behavior of very young infants is to look at something that's novel. That's how they learn about the world. So if the baby remembers the original face, he should look significantly longer at the novel face. And that's how we can get a measure of the baby's recognition memory. We also get a lot of information about the baby's attention and information processing speed from this task, because we look at things like the number of times that the baby glances back and forth between the two stimuli. That's been shown to be a good measure of visual attention. The baby's paying attention, studying the stimuli, processing the information. And then we also look at the total looking time of the baby and the length of each individual look, and that gives a lot of information about information processing speed. And it's actually been shown that babies that look for shorter periods of time have higher IQs and better executive functioning later in life. And it's believed because they are faster processors and they're processing the information more quickly when they're looking at these stimuli. So now I'm going to tell you a little bit about tasks that we're using to test babies when they're a bit older, at 4 to 5 months of age. And at this age, we're focusing on looking at cognitive tasks that are sexually dimorphic, that boys and girls tend to perform differently on. And the reason for this is that the chemicals we're looking at are all endocrine disruptors, and sex steroid hormones are very important for brain development and for mediating the differences between how males and females perform on cognitive tasks. So we wanted to take advantage of these types of tests to look at whether endocrine disruptors are influencing brain development. So we're doing two tasks. One's a spatial reasoning task called mental rotation, and boys typically outperform girls on this task. And this has been shown in older children and adults as well. And the other one is a physical reasoning task that was developed by Renee Bayer-Jean, who's a member of our research group. And on this task, girls typically outperform boys. So this is the setup that we use for testing older babies. When babies are a little bit older, we can actually use an infrared eye tracker to track their eye movements. And this is really nice because we can get really precise data about where the baby is looking when they're viewing stimuli. So here the baby's looking at a large screen, high definition TV, and mounted in front of the baby is the eye tracker. So babies sit on their mom's lap and just watch this display. So this slide illustrates the physical reasoning task that I was talking about. So there are two different types of trials that babies see on this task. In the first one, illustrated in the top brief video that I'm going to show you, a glove comes out and places a block near a wall. It's not supported underneath, but it seems to just be suspended there in space and it doesn't drop. In the other type of trial, the same glove comes out with the same box, but places it on the floor next to the wall, so it is supported. And what Renee has found in her work with babies at this age is that when you show girls the trial, like the one that I illustrated on the top, or what we call the magic trial, They tend to study this for a long time, indicating that they're surprised, that they realize that when a block isn't supported, it should fall, or they understand about gravity. Whereas boys don't tend to be surprised and don't look longer at that. They look equally at either the supported or the unsupported box. And on the right side of this slide, I'm showing some examples from the eye tracker. And this is just to show you the kind of data we can get and to show that, yes, when a glove comes out in the upper part of the display in places of blocks, the babies are focusing their gaze on the glove and then looking at the box where it's placed, and have very few fixations in other parts of the visual field. Whereas on the bottom, when the block is placed at the bottom, that's where babies are putting their attention. So, as I said, we're using this sexually dimorphic type of task to look at whether chemicals, particularly phthalates, which are anti-androgenic, might be influencing cognitive function. So it's interesting to think a bit about why this difference exists. And actually, it's believed to be because binocular vision develops earlier in girls. What this means is girls can have better depth perception and see a little bit better at earlier ages. So they actually learn about the physical relationships between objects earlier. It's not that boys won't figure this out soon. If you test them again a month later, they'll also be surprised that the box isn't dropping. And the slower development of this type of learning in boys, or the visual system in boys rather, is mediated by prenatal exposure to androgens, which actually slows down the development of the visual system. So we're interested in exploiting this type of task to see whether endocrine disruptors, especially anti-androgens like phthalates, could affect this process and disrupt the normal differences that you'd expect to see between boys and girls. At 7 to 8 months of age, we're doing a battery of tests with infants. We're looking at memory and attention using a very similar kind of task to the one that we used in the very young infants. In fact, this is a more typical age where this type of task is used. And this will allow us to look at the continuity of memory and attention and processing speed from very early infancy to 7 to 8 months. We're also going to follow up this testing with parallel ERP testing in 50% of our infants, and I'll talk more about that in the next slide. And then lastly, we're doing a theory of mind task at this age, which assesses the infant's ability to understand the point of view of others. So for the ERPs, our infants are going to Dan Hyde's lab. He's another developmental psychologist that's working with us on this project. The babies put on an EEG cap, as is shown here. And then they simply sit and watch a stream of faces appear on a computer screen. And many of the faces appear over and over again, so they become very familiar. And then occasionally a test image that is novel appears. So you can see that this has many parallels to the visual recognition memory test that we're doing in our lab. And then Dan can compare the responses of the baby's brain waves to the familiar and the novel test images. And by looking at the different scores between those, he can get a lot of information about how the baby is processing visual information, and also information about attention and memory that we hope will provide some clues to the underlying functions that are related to the cognitive tasks that we're looking at. And then lastly, I want to tell you about this false belief task that we're doing. And as I said, this is looking at social cognition or theory of mind. In the past, it's been believed that children really didn't have this kind of thought until around 4 years of age. But there was a very interesting paper in Science a few years ago where a group of psychologists did a task very similar to this one and showed that quite young infants actually can understand that others see things differently. So what happens in this task is there's two different kinds of trials. In the first one above, which we call false belief, the bear comes in with a ball. He puts the ball down on the table, the ball rolls behind a screen, and then the bear leaves the room. And while the bear is out of the room, the baby sees the ball roll away from out from behind the screen and go off the table. So it's no longer there. Then the bear comes back and the screen drops and there's no ball there. So in this case, the baby has seen something different from the bear. The baby knows the ball isn't there, but the bear doesn't. In the situation below, which we call true belief, it's very similar, except the ball rolls away while the bear is still there. So both the baby and the bear know that the ball isn't there when the screen drops. And what's been found is that babies look significantly longer in the false belief situation, which suggests that they are waiting to see what the bear will do because the bear doesn't know that the ball isn't there and should be surprised. So this is an indication that they understand that the bear has a different point of view than they do. So we're very interested in using this task because social cognition is an aspect that's very important in autism, and the fact that you can actually assess it at 7 to 8 months of age could be one of the important things in maybe early diagnosis of risk for autism. So that's all I wanted to cover today, and this last slide just reminds people that we have 4 students and postdocs from our group presenting posters, so please visit the posters and hear more about our work. And thank you.